As I said, Rachel Bearon is the second speaker. She's giving a talk on the impact of elongation on transport in shear flow. And please, the floor is yours, Rachel.
Thank you. And thank you for the kind introduction to this meeting. The talks I've been able to attend so far have been very interesting, and I'm looking forward to hoping to catch a few more over the week. So let me move forward. I'm going to share with you a couple of pieces of research which I hope will be of interest. The first involves swimming microorganisms in flows and looking at the effect of elongation on that. It was motivated by some fairly recent work looking at turbulent simulations, and that's work with this is work with Mac Durham from the University of Sheffield, and we're just in the final stages of getting that work revised for phys fluids. The second part of the talk again is about elongated swimmers, and in this case it's a little bit older work that I want to present, and apologies if there's overlap with Ganesh's talk. I wasn't able to attend his talk on Monday, and I know he's done some very nice work on this. So as I say, apologies in advance if some of it looks a little familiar, but hopefully it will still be of interest. And then there's a couple of more recent developments on that, which perhaps will be newer to those of you who are familiar with Ganesh's work. Okay. So we've seen these before, Jeffrey orbits. This is our starting point for Stokes flow. Elongated particles rotate at a non-uniform rate because of their elongation. So we're rotating because of the vorticity in the fluid, and we have the shape parameter alpha, 0 for a sphere through to 1 for an infinitely long rod. And here's vorticity, is the rate of strain tensor. Just want to highlight to those of you who may be less familiar with Jeffrey orbits, the fact that this non-uniform rotation means that cells spend, elongated cells spend longer aligned in the direction of the flow. So in this case, in this vertical direction. And this will become relevant in several of the examples that I'll be talking about. So the first kind of half of the talk is about looking at how elongation affects plankton in turbulence. And we're looking at a particular type of plankton. So unlike in Klaus's nice talk, where he was saying there's no kind of external forcing, in this case there's gravity. So we're going to be looking at gyrotactic plankton. A classic model for this is the idea of plankton being bottom heavy. So here's a little cartoon where the cell mass density distribution is not uniform, which means they end up being bottom heavy and therefore tending to swim upwards in still fluid. So the shape aspect is motivated by various examples of swimmers which are not spheres. So here's a couple of experimental images of some beautiful plankton, which obviously have these long and slender shapes. It's also got application, the maths that I'm presenting is application to other types of swimmers, some of the artificial swimmers that perhaps are not just actuated, reoriented because of gravity, but magnetic fields, the equations for those are very similar. Also, one of the motivations in this paper that was published a few years ago was the idea of chain-forming plankton species. So here we've got a chain of dinoflagellates, which often form, the individual cells can form into a chain and then swim through the fluid. And this we can model as a rod-shaped particle, and therefore, sit in a similar way to some of these others species. Okay, do interrupt, by the way, if you have any questions. I've enjoyed seeing people jump in the previous talks, so please do unmute and speak up. So governing equations for these systems, we're going to be thinking about the position and the orientation of cells. So position X is determined by their swimming. So P is the orientation vector, and a non-dimensionalized swimming speed given by this parameter phi. Cells, of course, are also moved by the fluid, and we'll assume they're just affected at the local velocity. So here we're neglecting any kind of inertial effects, and it's just very simple model for the cells moving in the fluid, and there's no feedback on the fluid by the cell's motions. Cell orientation, as I mentioned before, we have the Jeffrey orbit term, the second two terms here, the reorientation due to vorticity and strain. Alpha is our shape parameter. And then this first term is where we capture the alignment with the vertical K. So the second non-dimensional parameter psi, which we might call, sometimes referred to as the stability number. So this is a non-dimensional parameter which represents the balance between the viscous torque that comes from the shearing motion of the fluid and the gravitational torque due to the tendency for cells to align with the vertical. So increasing values of psi represent increased strength of the shear and vice versa. And there's a diagram again indicating what's happening with these cells. So in still fluid, there's still that swim upwards. If you have a bit of shearing happening, their orientation to the vertical gets tilted over and they're swimming up at an angle. So here's a couple of figures from simulations in turbulence. And I want to just point out a couple of key features which we want to explore and explain through by examining very basic flow fields to see if we can capture and understand some of the behaviors. So on the left, we see different numbers of cells in the chain corresponding to different values of alpha, going from spheres of 0 through to infinitely thin rods of 1. And for different values of the stability number, which can correspond to increasing values of shear, we compute they computed the vertical component of the fluid flow. So this is how much cells are being transported upwards by the fluid, normalized. And what they find is that as cells get more slender, as the chain gets up, cells typically spend more time in flow that's going upwards in this turbulent simulation. So they have an enhancement in their upward transport because they're kind of hopping on an up elevator, transporting them upwards. If we look at the right-hand plot, here we're looking at psi increasing values of stability number, or effectively increasing values of shear. And in this case, we're looking at the vertical orientation. So this is their ability to actively swim upwards. So their self-propulsion, their orientation PZ. And the plots are for the spheres and the limiting case of very slender rods with the closed and open circles for different values of the swimming speed. So what I'd like to highlight here is if you say, let's take just the red line and we compare the closed circle, which are the spheres, with the open circle, the rods, we see that the rods, the slender cells, have much greater vertical orientation. So they tend to swim up more rapidly for these parameter values. So these two aspects kind of combined suggest that slender elongated cells have the advantage in turbulence of being transported up both through the upwelling flow, transporting them upwards, and because they're swimming up more rapidly. And we wanted to understand why this was. Now, digging back to some classic results back from the 80s, we can compute what happens, what the equilibrium states are when we put these cells in simple shear. I'll put some maths up and then show some phase portraits to try and explain this a bit more. So for if we have flow which is horizontal shear, so we're having flow, this is just in the horizontal direction, we're thinking of all just 2D flow, simple XZ, just in the 2D flow, the XZ plane. Then in horizontal shear, there exists a unique stable equilibrium. So this is pointing upwards, the swim is going upwards, and we compute exactly what the angle with the vertical, theta is the angle that the cells make with the vertical is. So there's an exact single stable equilibrium, and this can be calculated and is known as a function of alpha, the elongation rate. And it exists provided our sigma, which is a measure of our shear rate multiplied by the local velocity, the vorticity, is less than a given value. And I'll show some diagrams of this in a moment. And similarly for vertical shear, analytically, we can compute this case for all values of alpha. So in some sense, the job's done. We can explain our orientation aspect, and that was half of the turbulence. We need to do a bit more about the transport. But when we were digging into this, the results didn't fall out right as we expected. And it turns out that although these stable equilibrium exists, they're not globally attracting, which hadn't very clearly been spotted before. But before I go on to explaining that global stability, let me just highlight what's happening when we have what happens in general. And this is something that was well known already. But I'm just going to orientate you with some phase portraits to help you understand help me talk through some of the later data. So I'm going to present some works for vertical shear. So this is the flow going in this direction, and for a value of a moderate chain with alpha being 0.6. I think this is a 2-cell chain. I think that was alpha 0.6. So if we consider a phase diagram of the orientation space Px and Pz, and think of trajectories through that phase space, for the parameter sigma is 0.5, this value of alpha, we have a unique equilibrium point indicated by this red big dot, and all the trajectories will end up at that equilibrium. So it's just off the vertical, it's just tilted over slightly in the positive Px direction. And if we plot it as a function of time, regardless of where you started with these different stars, you'll end up at that equilibrium point, just a simple equilibrium. And if the shear is greater, if we rank up the stability number times that vorticity, then what we get instead is the shear is too strong and the cells tumble over, head over tail, tumbling along. And that's exhibited by these orbits in the phase portrait and this periodic solution in T. Just to finish pointing out the phase portraits, these lines, the red and blue lines, correspond to the null lines in that dynamical system. So low shear cells go to equilibrium, high shear, they tumble. And this was known, this is not new, but just to familiarize yourself with the diagrams. So one of the kind of new results that we found when we dug into that is if you have vertical flow, so where the flow field is a linear shear in this direction, there are regions of parameter space when you're elongated where this equilibrium, this red spot at the top swimming upwards at an angle, is not globally attracting. So from the phase portrait, what we see is that if your initial condition is to the right of this red nullcline, you undergo orbit solutions. So there is this kind of tumbling, as it were, this orbit behavior, even though the parameters permit an equilibrium solution. So tumbling occurs more than, say, you might expect if you're just looking at where equilibria are feasible. It only happens for the vertical flow. The analysis for the horizontal flow, it's always globally attracting. And to start off, when we were doing this work, we thought this was a nice new classic result. It does turn out that there was a piece of work back in the 90s where they did actually prove this, but it doesn't seem to have been picked up very much by people working on a kind of Plankton and gyrotactic applications. So let's see what that means in terms of transport. So first of all, let's look at the horizontal shear. So this is the case where equilibria are globally attracting. It's the more straightforward case. So the velocity is linear shear. And we're going to have a few of these diagrams. So let me talk you through what the plots are. So we're looking on the bottom as the x-axis is psi. This is increasing values of shear. And on the vertical axis is the mean horizontal component and the mean vertical component. So this PZ is telling us how much they're swimming upwards. So that's quite important in terms of the vertical transport. Although PX will be also somewhat important later on. But let's focus on the PZ plot, first of all, on the right. And the different colors represent different shapes. So blue are our spheres with alpha 0. Red are the n=2 chains, alpha is 0.6, and increasing chain length. Green is 4, black is 8. And the red circles, sorry, the open circles are the analytic expressions for what this mean PZ should be. And the lines are the results from doing a large number of numerical simulations, individual-based simulations, deterministic simulations, but from a random starting initial conditions. And the bars at the top tell us the region of parameter space psi where we expect equilibrium to exist. So as you increase the elongation, there is a larger range over which an equilibrium exists. And this manifests itself, say, when we get at these larger values of psi as there being advantageous to be elongated. So if we look at psi as 2, for example, we see that where the blue cells, the spheres, they are tumbling away, there's no equilibria, and the mean upswimming is 0. As we increase our chain length from red to green to blue, black, we see an enhancement in the up vertical swimming. Yeah, I think the bottom plots are not so important to discuss for now. When we look at vertical shear flow, so this is the vertical shear where there isn't the global attracting equilibrium in all cases. The picture's a little bit different. If we look again at the PZ, we can have the solid lines are where an equilibrium exists, an upward equilibrium, and the equilibrium and whether it's globally attracting or not. So the dashed lines are where the equilibria is not globally attracting. And so here in this dashed line, depending on our initial conditions, some will go to the globally attracting and others won't. Massimo, have a question.
Just a curiosity. So to visualize what this means. So you suppose you do an experiment like the Kessler YouTube experiment of the 80s. So essentially, if you do it with elongated, rather elongated gyrotactic swimmers, what you would expect is that some of them will go up and some will remain somehow trapped at a given height. Those that tumble. I mean, is this that would happen, or depending on the initial condition, they will be trapped?
Let me think. So,
or not, because there is a transport by the fluid, maybe, I don't know.
Yeah, so I think I need to think a bit more carefully about what we'd expect the consequence in the pipe flow, the Kessler's experiments, and how much of the rotational diffusion would affect things. Whether you can get, so I think from an individual point of view, you would get knocked around with rotational diffusion and may attain that upswimming vertical equilibrium some of the time, but then you might get knocked off it and back into the orbits. And when you look at the orientation distribution of the population, you'll get this sort of, you'll get this bit of a mixed distribution. So I think I'd need to think a bit more to find out, to think exactly what I'd expect this modification to be, this non-uniqueness. But I'll go away and think of some more. That's a great question. Thanks, Massimo.
We can discuss later. Okay, thank you.
Okay, so what we see here is the circles are the equilibrium prediction for upswimming. The squares, if you can see the squares with the black, this is the modified equilibrium upswimming, which takes into account both the combination of that global stability and also the average behavior of the tumbling cells, which have no net zero upswimming. So if we randomly distribute cells around the unit sphere to start with, a proportion of them will have near zero net upswimming. And again, we see the effect, although it's a little bit less obvious, or maybe with the very long ones it's obvious. So say when we're at size 1.5, the very elongated cells, there's a big enhancement of their upswimming, mean upswimming, with this black line here. Okay. Yeah, so this just to highlight this bottom one at 1.5, this slightly confusing figure down at the bottom. What this is showing is cells which have attained an equilibrium are in plot D, and there's a large spike of cells that are swimming upwards in the black one. Oops, sorry. So a proportion of the cells at 1.5, which are long and elongated, are swimming upwards. If we extract out those and plot the distribution of all the other cells, we see all the other cells are orbiting at 1.5. So the kind of PDF of distributions is a symmetric distribution for each of the different colors. So, but then, and there's also these black cells which are orbiting. So there's this combination of a bunch of them swimming directly upwards at the equilibrium and a bunch are orbiting. Okay. Okay, so how are these cells transported? Horizontal shear, the only way they swim upwards is by, the only way they get transported upwards is by swimming upwards, because there's no vertical fluid flow. And generally, elongation improves and increases their upswimming. When you're in vertical shear, you've got two parts as to what transports you upwards. You've got your swimming, but you've also got the vertical component of the velocity, the fluid velocity, and that changes depending on where you are horizontally. So we need to think about the horizontal component of swimming, because if you've got a high horizontal component of swimming, as you do for the spheres, the blue line's the highest, you will be swimming into the region where the fluid's going downwards, and that will transport you downwards. And if you're trying to swim upwards, obviously that's a disadvantage. So elongation is good for both reasons. It improves your upswimming directly, but it also may suppress your migration into the downwelling flows. The final thing on this transport is to do with accumulation. So if we have, rather than linear shear, if we have some spatially varying shear, some Kolmogorov flow, what we can see is some sorting occurs according to the shape of the object. So when we have higher values of shear, say psi is 1.5, we can have, or the stability parameter, we can see layers of the blue cells, the spheres, emerging. Whereas if we have a higher shear, we can pull out layers of the red two-chain cells. And I don't think I'll go through that peak, but the second peak, this sorting behavior, where that comes about from is can be explained by some nice previous work that was done on spheres. And what I'll try and talk you through is what's happening. How do we get this accumulation of spheres at a certain height in the water column? So I'm jumping to a phase portrait again here. This time I've got the horizontal component of the swimming, px, against the height, z. And there's a nice constant of motion here. And if we think about a cell trajectory, what we find is it's following this black line. So its swimming orientation, px, is changing over time because it's being rotated by the fluid, but we end up accumulating just below this red constant here, line kind of manifold here, and accumulating at this blue point. So this is the point at which the cells tilt over and are swimming horizontally, so they don't swim vertically any further. And this accumulation point lines up with where we see the cells accumulating. So what's happening is there's a region here in the fluid where the shear is not too strong and the cells can swim upwards through it. And then there's a region here where the shear has got too high and the cells are tumbling. And this only happens for the blue cells in this example, because the highest shear in this example is 1.5. Now at 1.5, we see that the blue cells will be tumbling, whereas the other cells are still actively swimming upwards. So it's only a kind of lid, it's only a tumble zone for those blue spherical cells. If we up the psi to 3, we can now see that the reds will tumble at this peak shear zone. And we see an accumulation of the reds because below this region, the red cells have been able to swim up. So they've been able to swim up through this region where they do have an equilibrium. They've been able to swim up and then they tumble and accumulate. Whereas the blue cells show less accumulation because they've been tumbling over a larger region, all the way from where psi was 1 to 3. So they're sort of this region in the fluid. Okay, so the first part of this talk I've now concluded, and just a couple of points, I guess, to highlight. Importance of cells being non-spherical, that elongation is generally improving upswimming, suppressing migration into the downwelling flow, and we can get these shape-dependent horizontal layers. But I just wanted to flag up that this stability number psi and the speed phi do also vary with shape, whereas I've been assuming those are independent, separate parameters, and that's something that needs a bit further examination to really fully translate this into practical applications. Bernard, I think you've got a hand up.
So I just wanted to say, you contrasted the behavior and turbulence with your model, with a shear flow, and the shear is degenerate. The Jeffrey dynamics is very special in the shear. because alpha equals to 1 particles align, which they don't otherwise do. And so the question is, how generic are the results? So Kristian and I studied some years ago the same questions, but in a random velocity field. And we found that the particles, if the gravitaxis is quite strong, they tend to align in this, aggregate in the sinks of the transversal flow, which are actually the shear flow, shear, and then if you have a very, very slender particle, the shear rotates it like this. And so and then it crosses the line to the upwelling region. And that would also explain why, the particles tend to sample the upwelling regions when they are very slender. And I think that's probably a connection between, the special case you studied and the turbulence, which has generic linear flows. Is that consistent?
Yeah, no, I think your observation and comments are absolutely right. I think it's important and valuable to tackle and get to grips with some of the simplest flows, but you're absolutely right. In order to connect and translate that to the real situations and turbulent flows and more complex flows, it's important we recognise what those limitations are. So thanks for thanks for your comments on that. Completely agree with you. Yeah. Was there a second? Did Massimo have his hand up? No. Ganesh, did you have a question?
No, my hand remained raised by the previous question. That's why I lowered it now. Sorry.
Okay, fine. Good. If it's okay, I'll move on, change gears, and I'll probably go through the second part rather more quickly. So I wanted to talk a little bit about some work that I think Ganesh covered on Monday, and I've seen some other slides as well with people commenting on. And this is some because it feeds into some recent work we've been doing. So this is a question about kind of shear trapping and shear-induced migration of swimmers. So I think this slide's been shown before, where we have cells in a microfluidic device, and over time, cells are depleted from these regions of low shear in the center and accumulate at the regions of high shear nearer the walls. Just a quick little movie simulation, just to recap that this phenomena it doesn't require walls. So here's just an IBM where I've released a bunch of elongated cells, although it wasn't fancy enough to draw them as elongated, but they're undergoing the rules of Jeffrey equations in a doubly Poisson periodic flow. The red is just tracking an individual. And if you throw in a whole bunch of those cells and watch them accumulate, you see their accumulation is in the high shear regions, and they deplete from the low shear region. And the only components that are needed are some randomness in the cell orientation and elongation to get these accumulation in high shear regions. So the formalism that I've been working with is kind of Smolachowski equation, which obviously has a direct analog to stochastic differential equations or the active Brownian particle models that Klaus was mentioning. In this piece of work, we did include run and tumble motion, the velocity jump process, to allow us to explore chemotactic behaviors as well as rotational diffusion. So I'll just get all the slides up there. So what have we got? So it's a conservation equation for the probability density of cells having a certain position, x, orientation p at t time t. The first two terms are probably fairly familiar to people. We've got advection by the fluid, swimming, and translational diffusion. We've got our Jeffrey orbits plus rotational diffusion. And then the final term is where we capture the chemotactic behavior. So we've got this velocity jump process with a tumble rate lambda and a turning kernel k, and the chemotactic response is that the tumble rate, the probability of tumbling and changing direction reduces if you're moving up the gradient. So analogous to Klaus's nudging, we reduce the chance of tumbling if we're going up gradient. So this is the sort of the model framework that we're working with to explain and explore the shear trapping. Working in just a 2D setting, but based on the experiments and to simplify things, and looking initially to look at what happens as a steady solution for this 2D channel flow. So the governing equations that we saw a moment ago reduce to this set of equations where we can see just one physical direction y across channel, one orientation direction theta, which is the angle relative to the direction of the flow. We're assuming isotropic tumbles for our chemicals from our chemotactic behavior, so that's all quite straightforward there. This equation we are looking to solve numerically and do some analytic work with that. So one of the things that we looked at and didn't totally resolve, and I've got a bit of new work to show about this shortly, is if we've got this equation, what should our boundary conditions be? So when working with individual-based simulations, you know, we can specify boundary conditions, specular or random reflection. With these continuum models, I think there's still some open questions about what the boundary conditions should be. I'm just going to talk through the physics a little bit to try and explain what we did in that 2015 paper, which, you know, it turns out I don't think was completely accurate and correct, but more later. So physically, if I integrate over orientation space, I get the cell concentration. If I integrate the full governing equation over orientation space, I get what looks to me like a standard flux equation. And this is physical space. Therefore, I have a cell flux J, which I have obtained from the governing conservation equation. And the only thing I think I can confidently say physically is that this flux has a zero component into the wall. I don't want a flux of cells through the walls. So this integral condition holds. And I'm happy up to that. Problem is that that's not a sufficient condition to uniquely find a solution of that equilibrium. So in the 2015 paper, we picked something and we picked that integrand was zero at every value of theta. And we chose that as a, you know, a consistent approach. But I think that does some artificial boundary layers, which I'll mention a bit more later. So what we got out of that is solving that numerically. Solving that numerically. The green is the numerical solution. The blue is an approximate asymptotic solution, which I'll mention briefly, and the red is some experiments. The asymptotic solution that we obtain for small epsilon is that the density is proportional to e to the chi y. This example, chi was zero in the zero chemotactic case, divided by the mean square cross-channel swimming speed. And that approximate solution basically came about by neglecting translational diffusion, estimating this split between psi being able to be the cell density times the orientation distribution, and this leading our, and when we substitute that in, we can find what the leading order equilibrium orientation distribution is at each position in Y. Taking the first moment of the steady governing equation gives you this expression for what n is, and you can interpret n as being inversely proportional to this mean square cross-channel speed. So this is a little bit of a hand-wavy asymptotic approximation, which worked quite well for fitting the data and had a reasonable interpretation that you get accumulation where your cross-channel swimming speed is low. So that was, we were reasonably happy with that. So the sort of basic mechanism that we and others have discussed is this fact that when you have a cylinder shape, you have this non-uniform rotation, you get a peak in the orientation in the streamwise direction and reduction in the cross-channel swimming. I think that's slid a little bit, that picture. This is probably better, this diagram here. So that was nice and closed the problem a little bit, but there was a couple of problems with that, and I will just talk those through in the last bit of time to then identify some of the other work, more recent work. So one of the problems is that the approximate model didn't capture very well the effect of shape. So the approximate asymptotic solution here on the left, when compared to the full numerical solution, and we've got increasing values of beta from 0 to 0.8, we see particular discrepancies at large Peckley numbers between the full numerical solution and our approximate solution. Particularly, we don't get these double peaks in our approximate solution. So we're not capturing the behavior fully. But as I say, Ganesh probably talked a bit more about this work earlier this week, and there's a beautiful paper that came out, I was going to say last year, but we're already in March, so the year before last. And they did this asymptotics properly and very carefully computed all the details of the shear-induced migration, and were able to identify this separating region where you get the high shear trapping and where you get these double peaks. So my intention after that 2015 was to work on this and really get to the bottom of it, but thankfully Ganesh and his team got there sooner, and so this very much feels a very authoritative and really good paper explaining this. We also did some other work. So Lloyd Fung is a very talented PhD student with Yongwan Huang, who I discussed this problem with. And we just got a recent paper published about how we can get local approximation models for biased active Brownian particles in flowing suspensions published. And the maths in these couple of slides are a little bit heavy, but the basic principle about this is we're using a slightly different asymptotic approach to some of those that have been used. And what the approach allows us to do is develop the macroscale transport models for quite arbitrary flow fields. So this lovely paper was very much quite specific to channel flow. And there's been some other nice examples of papers which are sort of good for specific flow fields, whereas this approach we think should be more general for a wider class of flow fields. So let me try and talk through a little bit of the details of this. So the idea is that from the Smoluchowski equation, we can obtain an exact expression for the evolution of cell density, the NDT. And there's quite a lot of terms in here. So there's obviously there's the advection by the fluid, velocity u. There's then a family of additional drift terms here. or multiplied by this PES. So these are additional drift terms that are associated with the behavior of the cell swimming. There's then a regular diffusion, just Brownian translational diffusion. And then there's a couple of diffusion terms that arise because of the cell's randomness. So going into just a little bit more detail about these terms. So this PG, average motility from homogeneous solution of the orientational dynamics, VDT, this is this interaction between orientational and steadiness, VU to do with passive advection and so forth. As I say, there's quite a lot of terms in here that are broken down. And this is exact. Now, an exact formulation computationally is no better than just solving the full Smoluchowski. So you might think, well, what's the point of that? So then the next stage is to try and work out, well, can we approximate any of the terms in this? And when are those approximations valid? Because if we can get an approximate solution for the cell density, we can reduce the dimensions of the system and have an improved computational approach. And also we can try and understand what's driving some of those drift and diffusive-like behaviors from linking the microscale to the macroscale. So if we look at a test case, such as the one analysed by Ganesh, so thinking of pipe flow, steady pipe flow, no Brownian diffusion, the exact equation looks like this top one. As I said, in some sense, there's no benefit over solving the full Smoluchowski because all these drift terms and so forth will require quite a lot of calculation. Then the new approximation under these assumptions is given by this equation here. And in this case, the various terms in the formulation, the dxx and the vxgc, can be solved more efficiently. And here is some example of the calculations of those for a non-spherical weakly gyrotactic organism. And then, as I said, what's the advantage of this? It can then be valid. Oh, Massimo.
Yeah, excuse, can you come back? Yeah, I haven't understood the approximation. Can you spend a couple of words to explain the?
So it's an asymptotic approximation where we're going to be assuming that the PES is a small parameter epsilon. And so this will be looking, remind, PES, it will be a similar approximation to the asymptotics that I was doing a moment ago in terms of assuming that the swimming speed, a diffusive approximation where the correlation length and timescales will be small relative to some larger scale processes. I'm now wishing I had the paper right in front of me to give you a concrete example.
Yeah, okay. Don't worry. Don't worry.
It's a very similar type of scaling as that previous one, but as I'm sure many of you will be aware, you get a super student who goes with it, and they know it so much better than you. So I wish Lloyd was here.
Okay, don't worry, don't worry.
Okay. So yeah, so here's an example for more complex flows where it's been applied to. Okay, the second problem, this is the last thing I want to mention, and I wanted to include this because this is some nice work that Smita, who's joining us this week, who's a postdoc in my group, has been working on, and this tackles and explores this question about what I said about the non-uniqueness of that boundary condition. So we previously did some work where we just set that to be 0, and we said it's not necessarily the best one. So here's just a couple of results which haven't been published yet. We're just working on this at the moment, where we've been exploring some different boundary conditions. So here's some results where on the right, this is the orientation and position phase, kind of a 2D histogram, where we've done an individual-based simulation. We've specified specular reflection on the walls, y is -1 and y is 1. So the cells are doing their stochastic kind of standard stochastic simulation, where you've got the white noise for orientation, reorientations, and this is the accumulation that emerges from that. And what we're finding and what we're exploring in detail is that it appears that this very, very well fits with a doubly periodic Poisson flow. So as shown on the left, this is solving the continuum equation, but doing it in a doubly periodic flow field. So in some sense, we don't have to impose any boundary on the wall. It appears naturally, as it were. This doubly periodic Poisson flow has been used in simulations before, but we were wanting to explore how well it matches specular reflection, and it seems to be doing a pretty good job. And then this approach we can use to explore other boundary conditions. So yeah, the good matching here is a bit more detailed. So this is now showing the cell density. So this is summing up over the different orientations, cell density against the channel width for different values of the Peclet number. And on the left is the IBM. So purple is the smallest Peclet number, and increasing orange and blue. And this is the continuum with a doubly periodic Poisson flow. And we're getting very good agreement. There's a little bit of a dip at the high Peclet number with the near the wall, and that's a numerical artifact, which basically needs incredibly small DT time steps for the stochastic to avoid that. But other than that, it's agreeing very, very well. And then we can explore different boundary conditions. So here on the bottom, we've looked at having a random reflection. So rather than specular, we just bounce off the wall and come with some random U direction. And this is the profile that we get in that situation. It doesn't change too much the bulk behavior, but what we do get is these peaks near the walls. And if we're to match that to a continuum type of model, then our sort of natural assumption is that this should match most closely with a constant boundary condition. So we just impose psi to be uniform in theta at the wall. And we are capturing some of this boundary peaks and accumulations with this continuum constant boundary condition model. So this is kind of work in progress, which I hope to be able to share with you. And Smita is here, so if you have questions on that, you can ask. She'll be able to help you with that. Okay, so I think that's everything. So this second part was highlighting how we can explain this high shear, but there were quite a few challenges and problems, and I think meetings like this, where we have a chance to collaborate and discuss things and get new ideas, are really helpful to progress on some of these problems. So thank you very much for your attention.
So thank you, Rachel, for your talk. Are there any questions, curiosity, or? Okay, Ganesh, please.
Yeah, thank you, Massimo. Nice talk, Rachel. I had a couple of questions. One is actually more along the lines of response to, or maybe adding to what Bernard said midway through your talk. With regard to he obtaining the same results from a Gaussian random field, and as opposed to your considering a Kolmogorov flow kind of a profile. And I was wondering if it was a question of the shear that a particle or a swimmer sees. Is it the mean large-scale shear vis-a-vis the fluctuating small-scale shear that is inevitably present because there is a certain level of turbulence? And it actually matters as to which is larger and which dominates the orientation dynamics, which might differentiate the limit in which Bernard might work, where the small scale shear isn't limited and degenerate like the simple shear configuration that you considered. But in the other limit, the dominant unidirectional shear might overwhelm the small scale random shear. And the question that I had was about the last couple of slides where you had imposed boundary conditions. I was wondering if you have looked at the degree to which the boundary conditions penetrate into the bulk, so to speak, and if that boundary layer sort of a structure that might result from using one boundary condition vis-a-vis another, how does that boundary layer thickness depend on the flow strength, like a Peckley number? Thank you.
I'll comment on the second first, I think. So yes, that question really intrigued me when we sort of set off, because understanding the impact that the boundary condition has on the bulk, and vice versa, the bulk on the boundary condition, I think is absolutely crucial. So we did. I'm now thinking of the draft of the paper. So I might call in on Smita. Smita, when you looked at different Peckley numbers, I think you did a bit about looking at what the thickness of the boundary layer was, and it does a bit depend on Peckley number. I mean, in the extreme case, if you have no flow at all, you can choose all sorts of different boundary conditions that satisfy no flux, and they will very much determine what's happening throughout the region. But when you start turning up the flow, you get much more bulk dominated by the flow behavior. Smita, did you want to add any more on that boundary layer and the influence on flow?
Hi. Yes, so we certainly looked at the thickness of the boundary layers for the case with the pointwise boundary conditions. So where we said that the flux has to be equal to 0 for each and every orientation. In that case, we certainly found that the boundary layer thickness becomes thinner as you increase the Peckley number, and therefore have shear effects dominating in the bulk.
Great, thanks, Smita. I'm glad I was correct on that one. So I think understanding that, Ganesh, as to how important the boundary condition is in determining the bulk, but then also how important the bulk is in influencing what happens at the boundary is going to be really important for sort of applications that we've been thinking a bit about, things like early stage biofilm formation. So understanding what's happening in the bulk influencing the orientation that cells come and approach and hit the boundary is going to be quite important. The first question, this about the different scales of shear that are experienced by particles, I think that's really interesting, and I need to address that. I think a bit. That's one of the things I think we need to address a bit more in our revision at the moment. And I think. My hunch is that a big aspect would be being clear as to what sort of typical values of turbulent kinetic dissipation and so forth are going to be valid for the particular cases that we're interested in.
Okay, thank you.
Filippo, Klaus, I'm not sure who had their hand up first.
Yeah, I think Klaus, you can go on with your question.
Thank you. Thank you very much. I have a question concerning the orientation. Is there a privileged orientation near or where the strong gradients in particle density are or do you not look at the polarization there? I understand they are tumbling all the time, but there could still be some gradient in average orientation or something like that. Can you resolve something like this?
So which example simulations?
In the shear flows, if the particles accumulate, I understand they are swimmers and they are tumbling in the flow, right?
Yes. So the
their swimming direction is tumbling
with or without the gyrotaxis. So with the gravity as well, or without? So in the first half or the second half, were you thinking of?
Oh no, I thought just in general it could be in both cases, but they are always tumbling, right?
Yeah. So the orientation distribution. So there's two examples. So in like these pictures here, where this is the orientation distribution. So theta is the orientation. So we can see how the orientation distribution varies across the channel.
Yes.
So we're keeping a track of that.
Yes.
So in this situation, the cells are constantly tumbling. So any individual will be doing a full circuit, but because they're not tumbling with uniform rotation, you get these peaks in orientation.
Exactly. And that would then that is already driven by the flow, but then it's modified if you make the particles active, right? I mean, if you compare a passive and an active particle, then this pattern is modified.
Absolutely, because they'll be swimming across streamlines and so forth. Because they'll be swimming across streamlines, and so they'll be sampling different types of shear. So yeah, the ability, the active nature will change that.
And can I imagine that when they are rotating faster and on one side and slower on the other side, then that there is basically also a net difference in diffusivity of the swimmer, like referring to the swimming, because rotating faster means that it's less persistent.
Ganesh, I think, Ganesh, were you about to jump in with a response on that? Or was it a different question?
No, I just wanted to add that I think with regard to one of Klaus's points, in the absence of gyrotaxy, there will be a nematic symmetry. I think so that the probability distribution of orientation P should be equal to the number of swimmers with minus P. So I think there will be a P minus P symmetry. But with gyrotaxy, that symmetry is obviously broken because the direction of gravity now breaks that symmetry. So there's going to be a polar kind of an asymmetry with gravity in there.
Yes, but without, you say it's symmetric. But as I showed, I mean, in my talk, when you have a more active and a less active region, then there is a net orientation at the boundary. But this was completely without any superimposed flow. So it's much simpler, the situation. But just by having the different activities, or like different effective diffusion coefficients, if you want, of these active swimmers, you get already some net polarization.
Yeah, so I think that, sorry, Rachel, go ahead.
Yeah, so I mean, it's always interesting talking to people with different backgrounds of explaining things.
That's my problem, maybe, yeah.
So I think going back to this, cartoon back on this right-hand side, this cartoon. So what we're having here is this question of shear trapping. I think is one of the things which that we're trying to get across. So in this situation, the cells are all doing their rotation, their active rotation around. If the cells were not active, if they weren't swimming, you would just get a uniform distribution of cells. They wouldn't generate these accumulations. But because they're swimming, because they're crossing streamlines, you end up getting these accumulations. So there's sort of, I guess, in some sense, an effective, reduction so a reduction in effective diffusivity almost, and these accumulations, or it's a kind of an effective drift that occurs.
Yes, and could you say that whether this goes like square root of the effective diffusion coefficients? That would then be my question, because that's what we see in the quiescent situation, that the density difference scales like the ratio of the square roots of the effective diffusion coefficients.
So
like the active.
Yeah, so the scaling, I'm just trying to think now. There is a scaling in there, but I'm now, because everything's been non-dimensionalized, I'm now, what is that scaling? So this accumulation, you can ignore the top part because that's just the chemotactic. So the effective accumulation that we get goes like the mean square cross-channel swimming speed, which I'm just thinking, oh, I'm wanting to say yes, and I'm struggling to think of exactly what it should be. This is when it would be nice if we were all in person, wouldn't it? Because I could scribble away on a piece of paper over lunch and get back to you. But I think there should be. Filippo, did you want to, maybe someone else wanted to chip in? Or Ganesh?
Yeah, sorry if I interrupt. We are a little bit out of time, so I will say now there is the break. We will start again in 15 minutes, but so at 11:45 European time and 4:15 I think Indian time. But I mean, I think that you can stay here, continue the discussion, and for all the others, we will be back in 15 minutes, if you agree. So Filippo, if you want to go on with your question.
No, I think it's better if we maybe chat a bit with Rachel and so on, because there are several topics here, and so I don't want to expand more. But it was just a comment. So I think it's the right choice to close the session now, and we go on discussing maybe over our coffee. If someone is interested, they can follow the discussion. Okay. Thanks, Mattia.
Okay,
Yeah,
so
and thanks, Rachel.
I'm sorry, I realized that I was a bit the culprit for running overtime in my talk, and now also with a complicated question which was not well posed. No. I'm sorry for that. Yeah.
No, but it was the right choice of Matteo to
allow it to go.
Say that everyone is officially everyone is allowed to go for a coffee.
15-minute break.
And since we are more invested directly in this thing, maybe we can go on discuss. Now, my comment was, so thanks to everyone. So now my comment was partly on the alignment, because so if you start putting, for example, fluctuations, you have flow like turbulence or some random flow, and it seems like, okay, two things. It seems like orientation with the flow, with the mean flow, is quite robust, and so you still have symmetry breaking basically, and so you have polar orientation and polar ordering. The second thing that is quite interesting is that apparently, as soon as you depart from the very low diffusivity case, I mean, if you think of fluctuations of the flow, like small scale flow or something like that, as an effective diffusivity, as long as you depart from the small diffusivity case, you tend to lose very fast the accumulation, and still the orientation remains. So you keep the symmetry breaking in orientation, but you lose the accumulations quite fast. We did some work recently, and I can share the preprints with you, the preprint with you if you want.
So there's no simple lockup between orientation and density.
Yeah, because basically you tend to, you tend to orient with the flow, but you still have effects of the flow that move you around, that cause a space diffusion, right? Just purely. And so yes, but still orientation works because it's more of a local effect, and you have the effect of the mean shear and so on. And so just to complicate it.
It's so many contributions, it's hard to understand which ones are the dominant.
If I can add only one thing to what Filippo said, I mean, what we noted also before, also within homogeneous isotropic turbulent flow, you have this alignment with the local flow. And essentially, we understood mostly through the work of Kristian that worked out the analytics that, I mean, it is essentially whenever you swim, you break the symmetry, you break the Fourier symmetry. So if you are not swimming and you're with the same elongation, you are like a kinematic with respect to the velocity. You still have alignment, but is in both directions. While as soon as you swim, you break this symmetry and you have a polar order. Of course, on average, not constant. Of course.
Yeah,
exactly.
Yeah.
So it's very similar to what you were somehow saying for the active particles. I think. I mean, even.
Yeah. Just much more complicated because you have all these many effects on top of each other, right? That's what I got from Rachel's results. And I asked myself, is there the simple bit I described is somewhere hidden in there, but it's hard to see it. And,
the interplay between motility and the fluid transport is quite, I mean, strange. I mean, can
has many aspects, more than, it's not just.
Yeah, for example, Rachel was showing, you know, that when if you're spherical, even if you want to swim upwards through gravity axis, if there is a flow, you tend to aggregate where there is a downflow, which is counterintuitive somehow. No, I mean, you can have a very complicated mechanism around. Yeah.
I would like to look a bit more, Klaus, at your paper, because I think it would be nice to be able to make that connection, because it very much chimed, particularly about the passive active and looking at the distributions. I do think that there is definitely some connections there, and it would be good to try and work out a bit more what those connections are.
I have the feeling that it might be a super weak effect, because in our experiment it's a strong effect, but we have a passive and active, and then we have a Peckley number which is higher than 1, and so there is a big jump, and then there's a big effect. But in your case it's very gradual and it's not that strong, because they are always active, more or less active in a sense of a diffusion coefficient. So it must be much more gradual, and maybe it's completely overwhelmed by all the other terms in your equation. So that was my feeling a bit, but yeah, it's hard to get a feeling if you don't know the importance of the different effects, and I lack too much background, I guess. Yeah.
So, and if I can add an ingredient to the complication of the thing, there is also one aspect that when you have flow, the correlations are important. So the correlations, the fact that when you swim, you have correlations with the flow, right? Which is quite different from just noise. And so all the different cases pose different problems, right?
So I just wanted to make one point. I think at some point Klaus asked what if there was no swimming. So even if we had passive Brownian anisotropic particles in this flow, the flow that Rachel is considering, parabolic flow, there would in fact be a small inhomogeneity, just from the fact that the effective diffusivity along the gradient direction varies because of the orientation. So it need not swim for it to develop a gradient. It's just that the effect is so small for because of Brownian motion. So if the effective temperature were to be actually much larger than the thermodynamic one, then maybe one could enhance that effect. But it is there even without swimming in principle.
I understand. Yeah, that's good. A good remark. Yeah.
Sorry, I leave you for 5 minutes since you.
Yeah.
Later.
You think I could try to share?
Thank you very much for taking my stupid questions, and I have learned a lot. Thanks.
Likewise, really nice to meet you. And I'm sorry we're not being able to meet in person, but another time maybe.
Yeah, it will be very nice. Yeah.
Okay. Thanks.